Hello, I am Professor Sims, and this video is an introduction to compound light microscopy. This is the third in a series of 10 lab sessions held as part of my Laboratory for the Fundamentals of Microbiology course. If you're a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. The learning objectives for this lab include learning to identify the parts and the function of each part of a compound light microscope. You're going to learn to properly operate and care for a compound light microscope. You'll become competent in basic wet mount slide preparation. You're going to learn to observe various specimens using a light microscope and also using the Foldscope instrument. First, we'll talk about compound light microscopes. Microscopes in general are needed to view microorganisms such as bacterial cells, which are so small that they're measured in micrometers. A micrometer is 1 × 10⁻⁶ meters. Here you have a nice figure demonstrating the limits of what you can see with the human eye versus a light microscope or an electron microscope. The human eye, obviously you can see the human or hand or finger, but the limit here is going to be about the size or the thickness of a human hair. For things smaller than that, you're going to need some kind of microscope. A light microscope can see individual human cells, bacterial cells, some subcellular structures. And if you want to see something as small as a virus is actually 3 orders of magnitude smaller than a bacterial cell, and it's measured in nanometers. So for that, you're going to need an electron microscope. And an electron microscope, depending on which type you're using, and we talk about the types of electron microscopes much more in the lecture course, but some of them can see as small as a single atom. The compound light microscope, which sees within this range here, is generally what is used in a traditional microbiology lab course. Parts of the microscope are all defined here. Be sure you know the name and the location and the purpose of each of these parts. This figure here illustrates the parts of the scope. You have the ocular lens, objective lens. This is the nose piece that holds the objective lenses. The arm. You have coarse adjustment knob around the outside here, this big knob. And on the inside part is the fine adjustment knob. The base. The illuminator, which is the light source. The condenser piece, and then just below that is the iris diaphragm. This is the stage where you put your slides. These are the slide holders here. Again, just be sure that you can identify all of the parts and know how to properly use each part. As far as focusing and observing specimen, it is important to understand how the microscope works in order to be able to focus and view your specimen. So in a compound light microscope, you have light that comes from the source, the illuminator. It is first focused by the condenser, the lens in the condenser. Then it enters the objective lens, where it is magnified in order to produce a real image. So you start with a virtual image, and then you end up with a real image that you can view with the eye. The real image ends up being reversed, so it's mirrored upside down and it's mirrored horizontally, which becomes important to know when you're actually trying to move your slide around in order to get a better view. You have to understand that it is going to be the opposite, both vertically and horizontally, from how it looks like it is on the stage versus how it's going to look to you through the ocular lenses. Therefore, it's going to take a little bit of practice to orientate yourself when you're using the microscopes. You need to know the steps involved in using the 10x and 40x objectives, and also later on we'll talk about the 100x oil immersion objective. So first you want to turn the illuminator on. You start at about half power because it's very bright when you first start on 10x. You use your coarse adjustment knob to lower the stage. You set the objective to 10x. You place the slide on the stage, and you use the coarse adjustment knob to raise the stage so that you have a minimum working distance. The working distance is just the distance between the objective lens and the slide that you're trying to view. You will adjust the interpupillary distance, the distance between your ocular lenses, and that helps to make sure that you're not seeing double. and then you look through the ocular, rotate your coarse adjustment knob slowly so that you can get the specimen into view. And then once the specimen's in view, you use fine adjustment to focus the image further. And then you'll rotate the objective lens into the 40x position, and you'll refocus again using just the fine adjustment. With a light microscope, compound light microscope, you're only ever using the coarse adjustment knob on the 10x objective. Anything above that, you have to use only the fine adjustment. Mastering the light microscope instrument requires understanding and applying the concepts of microscopic field, parfocal distance, magnification, and resolution. The idea of microscopic field and parfocal distance is basically as you go up in magnification, your specimen that you're looking at, it's going to look very different. And the reason for that is not only because it's getting bigger, but because you're looking at a much smaller portion of the specimen. So your microscopic field, when you're on 10x, as you can see a much larger part of your specimen. But then when you go up to 40x, you're zoomed way in and you can see a much smaller part of your specimen. When you're on 100x, it's even way smaller than that, okay? So these are all the same specimen, just at different magnifications. The parfocal distance concept means that you should be able to move from one objective to the next without doing very much adjustment of your specimen. In other words, you don't want to move it around too much, and even adjusting the fine adjustment knob should be very minimal. You want the changes that you make between the objectives to be very slight, very minimal changes. Because if you lose your specimen, you can't see it anymore on 40x, you're not going to find it again on 40x. You're going to have to go back down to 10x, start over, and then go back up to 40x. Same thing for 100x. So finding a bacterial cell in 10x is like finding a needle in a haystack. Finding a bacterial cell in 100x is going to be like finding a needle in a stack of needles. It's going to be very difficult. And that's because not only are your cells getting magnified, but so is all of the white space around it. And if you move this slide a little bit too much to where you're focusing on the white space, all you're going to see is white in 40x. All you're going to see is the white space in 100x. So that's the idea of parfocal distance. As for magnification, you need to understand that magnification and resolution are different things, but they do work in conjunction. So your magnification is measured by the ocular lens magnification multiplied by the objective lens. So magnification, total magnification equals ocular times objective. Most ocular lenses are going to be 10x. The ones I use in my on-site lab are all going to be 10x. And the objective lenses are the ones that change. So those are the ones that are on the nose piece and you can rotate. Some scopes have them as low as 4x. For microbiology labs, I generally have students start at 10x. And then there's also 40x and 100x objectives. So if you're looking through a microscope that has the objective set to 40x and the ocular is 10x, then that means the total magnification is 400x. And magnification really is just making something bigger. If you want something to be focused, well, that is actually resolution. Resolution is the ability to distinguish between 2 points, a specified distance apart, but far enough for light to pass between them. So basically what that means is you can have something that is magnified very nicely, but it is blurry, or it's out of focus, or you really can't see the structures. All you can see is the color or the basic shape. In order for it to be clear and focused, you need to have good resolution. Resolution is determined by the numerical aperture of the objective lens that you're viewing your specimen through. The numerical aperture is a measure of the cone of light that can be gathered by the objective, and by using immersion oil, you can change the numerical aperture, so we'll talk some more about that in just a bit. But essentially all you really need to understand with this, you don't need to understand the physics of this, but you do need to know that the different objective lenses do have different numerical apertures, and that is what determines the resolution power of those lenses. For experiment 1, all you're going to need is a computer or another internet-connected device. You're going to navigate to the following URL, and you're going to complete a tutorial. It's a tutorial where it's going through the basic concepts of light, how microscopes work, how different types of microscopes work. So not only the light microscope, but phase contrast, fluorescence. I think it even dives a little bit into electron microscopes. So you want to click through all of the links or all of the steps there and watch all of the animations. Record your answers to the guided questions and the Lab Report 2 template. Also, here's just a little short clip of what this tutorial is going to look like for you.
Light is created when electrons within the atoms become excited by absorbing extra energy from their surroundings. The extra energy causes the electron to move up to a higher energy level within the orbital shells of the atom. Since atoms seek stability, the electron will return to its original orbit, releasing a particle or photon of energy in the process. A ray of light is a steady stream of photons traveling along in an electromagnetic wave. The discovery of the law of refraction in 1621 made possible the development of the compound microscope. The basic compound microscope consisted of two convex lenses in series, an objective lens close to the specimen, and an eyepiece lens near the observer's eye. Light microscopes may illuminate the sample from the opposite side of the lenses, known as transmitted light, or illuminate the sample from the same side as the image-forming lenses, known as reflected light. Modern contrast in biological tissue sections can be enhanced by staining the sample.
Next, we talk about the oil immersion objective, the 100x objective. So the key here is you're dealing with refraction of light. So you should have seen in the tutorial how important it is to gather light and how important it is to try to keep your light from getting refracted away when you're focusing. And the refractive index of your slides is about 1.56, whereas the refractive index of air is 1.0. So what happens here is once you get down to the higher objectives, especially the 100x objective, you need to really capture all of your light, but you do have light that gets refracted in air. So in order to try to help with that, when you're using the 100x objective and you're in a really high magnification, remember 100x objective lens, if you're coupling that with a 10x ocular lens, that is 1,000 times actual size. So you're getting into really high magnification, you need to really focus with all of the light that you have available. So what we do with the 100x objective is we use what is known as immersion oil. The immersion oil forms like a little bridge between the light source and the slide and the objective lens. And that way you reduce the amount of light that is lost. It's very important whenever you're using the oil immersion lens that you clean the oil after you use it. Don't want to leave it on the lenses. And you're never using immersion oil with any of the other objectives, just the 100x objective. So again, here's some special precautions that you are going to be using for the 100x objective. Again, you do need to know how to use all of the parts of the light microscope and how to care for the scope. Make sure you're never using coarse adjustment with the 100x objective. You only use the coarse adjustment with the 10x objective. You add oil to the slide by rotating the nose piece halfway between the 40 and 100x objective. Then after you've added the oil, you'll rotate the 100x lens into place. The lens should be touching the oil. So the oil is touching both the lens and the slide. For experiment 2, you're going to again need a computer or another internet connected device. You're going to navigate to this site here. You're going to complete the simulation and record all of your answers to the guided questions in the Report 2 template. This one is a pretty fun simulation. Here is a clip kind of demonstrating how to navigate this simulation.
Hello, Lab Assistant. Today, we'll view the gram-stained yogurt sample with a microscope to get an idea of the kind of bacteria contaminating it. To learn more about the purpose and techniques of gram staining, please take a look at the Gram Staining Virtual Lab. A short background about gram staining. Hans Christian Gram, a Danish scientist, devised this technique to let bacteria. We will use a light microscope with 10 times, 40 times, and 100 times lenses. The 100 times lens is an oil immersion lens.
Experiment 3, we're going to make one glass slide with the pond water specimen, and you're also going to be making a wet mount with using a piece of your hair. As for your pond water specimen, here are a lot of examples of what you might find in there. These guys over here on the left are all swimmers. So if you see things swimming around, you see things moving around, it's probably these guys. Amoebas move using pseudopods, false feet. Most of these others are using cilia or flagella. The paramecium here, these guys are just covered in cilia and they move very, very fast. These guys that have like one flagella at one end, they move a bit slower and they whip around, like they whip back and forth. Things like this and this guy here, they kind of swim around in circles. So you can see any or all of these things. It really just kind of depends. Your Spirogyra are pretty cool. They're rather large, eukaryotic, multicellular, and they are green because of all of these chloroplasts that are inside. Algae, you're going to see probably a lot of algae. It's greenish or brownish. In fact, algae are a good place to start looking for swimmers if you're looking for swimmers, because these guys like to eat the algae. So if you find a big clump of algae first, you kind of focus in around the algae, you may very well find some of these guys. Some of the observations: were you able to identify all the parts of the scope? Do you know how to use the parts of the scope? Were you able to observe specimen? Were you able to make your wet mount slides correctly? Were you successful in viewing your wet mount slides? Do you know why a cover slip is necessary when you're using a wet mount? That should become pretty clear to you when you're actually trying to view the wet mount slides. So I hope you really have fun with Lab 3. We're going to be looking at things that are on a larger scale as much as possible. When we get to some of the smaller things in Labs 4 and 5, you're going to see even more online simulations and stuff. But for Lab 3, you should be able to see all of these specimens. You should be able to focus them and have fun with it. Okay, well, thank you guys for watching. And don't forget to do the reading and check the description for more videos related to these topics. And leave your questions for me in the comments below.